My name is Liang Huang from Bio Research USA and Oregon State University, and I would like to thank the organizers of this workshop for inviting me, especially William for inviting me to give this talk. So actually, I'm not a biologist. I'm not even a computational biologist per se. I'm a computational linguist by training, and most of my previous work has been in computational linguistics, also known as natural language processing. But in the past, like 5 years or so, I've been gradually adapting more and more of my algorithms from computational linguistics to biology, and especially RNA folding, because these two fields, even though they're far apart on the surface, they actually share the same mathematical foundations. So I adapted my linear time parsing algorithm from linguistics to RNA folding, which resulted in LinearFold, published last year. Which is the first linear time algorithm to do RNA structure prediction, and it's orders of magnitude faster than previous cubic time methods. Cubic time methods are kind of similar to our classical CKY style parsers in natural language processing. And when the pandemic strikes, you know that the coronavirus has the longest genome among all RNA viruses. Its genome length is 30,000 nucleotides. So N is 30,000, which is much, much longer than any sentence in, let's say, English or Chinese. So the impact of my algorithm is much more bigger in biology, and especially because of the pandemic. And we also extend it to do the partition function of all possible structures. And that's a well-known or widely used concept in classical structure prediction, like conditional random fields, for example. Partition function, Markov random fields, but it's no longer being used very often in today's deep learning environment, unfortunately. And we also extend it to do conserved structure prediction, so that we can look at coronavirus genome and see which segments of the genome is well conserved among all the variations. So those parts are the critical parts of the virus and will be targets for drug design. So this is the first time that conserved structure prediction can scale to the coronavirus genome scale of 30,000 nucleotides. So those RNA viruses like HIV, Ebola, MERS, SARS, coronavirus require very fast, efficient algorithms. And it's lucky that I've been developing those algorithms from computational linguistics, and they suddenly have a lot of usage in this pandemic. And these are my students and co-authors, and we also have a great collaborator, David Matthews, from University of Rochester Medical School. But pandemic changed everything. Since the pandemic, I've been asking myself, how do you do something more directly to help fight this virus? So we're thinking about vaccine design, and a professor in Stanford Medical School, Riju Das, told me something about the principle of vaccine design and the major challenge there. So it turns out that mRNA vaccine design has emerged as probably the fastest and safest route among all the vaccine types. It's much faster for mass production than the classical vaccine type, which is the protein vaccines like flu shots. But it's still a long way to go, with many challenges remaining. Especially the most important challenge, arguably, is the chemical stability or instability, rather, of RNA vaccines, and because of that, the low immunogenicity problem. So you probably read the news that Moderna and Pfizer and maybe other mRNA vaccine companies need very, very low temperature, ultra low temperature, like maybe negative 50 Celsius, for the storage and shipment of those vaccines, which increases the cost. And also, you probably need larger doses because of low immunogenicity, and that would induce more side effects. And you can read it from Moderna's phase 1 or phase 2 reports. And our idea to solve this problem is to use some classical work from computational linguistics to design more stable mRNAs. And because of the stability, they would lead to also higher protein expression level, so that it will produce more antigens to trigger your immune response. So as we all know, there are 3 types of vaccines. Protein vaccine is the dominant one, and almost all the vaccines you have seen so far are protein vaccines, like flu shots. But it's very, very slow for mass production because you need a lot of eggs or cells to grow or replicate or cultivate the virus inside the eggs. So that's why you need a year for every year you need a long time to make a new flu shot. And it has also a small risk of infection. The DNA and mRNA vaccines are gene-based vaccines, also known as source installation instead of binary installation. So protein is like a binary installation. It directly installs the antigens, the proteins, into your body to trigger the immune response. But this DNA of mRNA vaccine injects not the proteins, but the genes, so that will be translated into proteins. So they are more like source table installation. And DNA vaccine and mRNA vaccine are much faster in mass production because you don't need eggs or cells. It's no risk of infection. But DNA has a small risk of integration to your genome because it has to enter your nucleus to be transcribed to mRNA and then translated here in cytoplasm to proteins. mRNA only enters cytoplasm to be translated by ribosomes to proteins or antigens. So it's probably the safest and fastest. But the problem, as I said before, is the chemical instability, because RNAs are mostly single-stranded. And because of that, it can easily be cut down in water or other molecules because hydrolysis or other enzymes are trying to degrade it, cutting down the single-stranded portions of the sequence, whereas the double-stranded pieces or segments of RNAs are more stable, just like a DNA, right? So DNAs are much more stable than RNA. So the question really is, how do you make this mRNA more like a DNA? How do you make it more stable chemically by changing the design of the sequence? And because of the stability, probably use higher amount of proteins or the antigens, so that will have better immunogenicity. So that's the question we're going to solve using computational linguistics today. So mathematically, the problem really has an exponentially large search space. So formally speaking, mRNA design problem is trying to exploit the redundancy in our genetic code, because each amino acid can be translated by multiple DNA codons or RNA codons, also called triplet codons. So there's 4 to the power of 364 possibilities, possible triplet codons, and only 21 amino acids. So each amino acid has, roughly speaking, 3 different ways to be translated, right? But if you list them down, and if you say, oh, this is the coronavirus spike protein sequence, and by the way, spike protein is the most commonly used vaccine target. So you want to kind of express or translate into this spike protein, which will your body will trigger or will recognize. So next time if the real virus strikes, you'll say, oh, I saw you before, right? So you say, oh, methionine, you have 1 choice, F, you have 2 choices, valine, you have 4 choices, blah, blah, blah. You times them all together. The product of all the choices, you got 2.4 times 10 to the power of 632 choices or candidates or sequences. So all these sequences translate to the same protein, which is the spike protein. So these are called synonymous mRNAs. And which one is the best? Well, according to the Moderna paper, you want the one that is most stable. In other words, the lowest free energy or the highest amount of secondary structure, because that stability also yields to high protein expression. But the question really is, how do you try to find the best one, the most stable one among this huge space? By the way, if you can enumerate them like 1 second a sequence, this will take more than the life of the universe. So it's definitely impossible to enumerate them one by one. So how do you do the search efficiently in this gigantic space? Well, it turns out that this is a classical problem that in computer science we have solved it in 1961. So this problem was given to us by Professor Rujudas from Stanford, who introduced us to this problem. But he thought this is like an NP hard, it's impossible to solve, it's way too big for combinatorial explosion. So he designed another game called Open Vaccine, which uses my linear fold, which I introduced in the first slide, plus cross-sourcing. So that he asked players to help design the vaccine and use my linear fold to score the energy, the stability. So my linear fold will report the stability very fast, in like 3 to 4 seconds, because it's the fastest folding algorithm. But I thought that would still be impossible for human beings to do a gradient descent in this kind of space. So human gradient descent might be more useful, more kind of intuitive than machine gradient descent, because machines are kind of blind. Humans have better intuitions, but this is still way too big for humans to search. So I would like to say use a direct optimization method. So how do we do it? Well, before I talk about technical details, I would like to show you the results. Well, so this is the wild type of the coronavirus. You can see that it has very bad shape. It has a lot of single-stranded areas, which can easily be cut down in water. So largely single-stranded, high free energy, and easily degraded. After about 10 minutes of optimization, we got something much better. As you can see, they're mostly double-stranded, and so it's no longer very easy to be cut down, and it has much lower free energy and much more stable in water or other environments. And you're trying basically making an RNA into a DNA, roughly speaking. So how did we do it? Well, here's the optimization problem. Here's how we treat this. So we treat each mRNA as a DFA. We treat each amino acid as a DFA. So, for example, valine is an amino acid, has 4 different choices of triplet codons. So we represent them as a DFA. Serine has 6, we represent them as a DFA. And then you concatenate these little DFAs for amino acids together to organize into a protein DFA. And this very long protein DFA encodes all the mRNAs that can translate to the same protein. For example, all these different paths in this DFA translates to the same protein. So they're all called synonymous mRNAs. So in other words, we have a very compact space encoding all the exponentially many possibilities. So that's our first observation. Our second the great ingredients of our work is to represent the folding free energy of RNA as stochastic context free grammars. That's very old stuff. That's stuff dating back from 1980s, and that's something I talked about in the very beginning. Why these two fields share the same mathematical foundation. But the next observation, which is new, is to realize for the first time that mRNA design problem is nothing but CFG intersection with DFA. Why? Because how do you find the best path in this, in the exponentially many possibilities in this DFA? Well, we superimpose the CFG, the context free grammar, on top of this DFA to do the intersection, and we know that CFG intersection DFA you got another CFG, a larger CFG, and that's a classical concept in the theory of computation and computational linguistics, which dates back to 1960s. And dynamic programming can solve it in cubic time, where n is the RNA sequence length. So that's very efficient. So that's the kind of eureka moment that we realized this problem is actually solved 60 years ago. It's just another classical problem. So we implement it, and the speed results is that you have 2 versions. One is exact search, the other is approximate search. And the exact search can finish in about 1 hour, and you lower the energy by more than 2-fold. And approximate search is much faster at 11 minutes, and the energy is almost the same, only like less than 1% difference. And as you can see, the design structures, wild type is very, very bad in energy and shape, and linear design is much better, much tighter, so that it's mostly double-stranded and much lower in energy, so it's much more stable. But that's only half of the story. Well, the other half of the story is that you have to consider codon autoality, where human cells prefer GC-rich codons, and other like organisms like yeast maybe preferring AU-rich codons, but different codons for the same amino acid have a distribution, have a frequency or probability. So for example, if you want to encode leucine, you would prefer to use CTG. So you would ask your design to be using the frequent codons more and more, so that you can increase the translation efficiency. So that's why it's really a 2-dimensional optimization. On the x-axis, you want the lower the better, because the lower the energy, the more negative the energy, the more stable the structure. On the y-axis, you want higher the better. The high CAI means codon adaptation index, closer to 1 meaning more efficient, using only the frequent codons, very frequent codons. And wild type is very bad in both structure and CAI, in both energy and CAI, because it doesn't have enough time to evolve in the human organism, so it still is very kind of primitive from the virus. It's just recently jumped onto humans. So, but if you do CI greedy design, then for each amino acid you choose the best or most frequent codon, and you got a CI of 1. And because human codons prefer GC richness, and GC richness gets lower energy or better structure, so you actually got a byproduct of lower energy. But it's only a little bit better, not too much better. And most vaccine companies are doing codon optimization, like here, like very close to here, which you get much higher efficiency and also lower energy and more stability. But the whole area left to this line is uncharted area. Nobody knows what's going on here, and there is also a kind of feasibility limit where above which it's impossible in feasible region. So lambda is a hyperparameter which controls the relative importance between CAI and MFE. For example, lambda of 0 means you only consider energy, no CAI, and lambda of infinity means CAI greedy design with no energy in consideration. With varying lambda, you got a curve, and beyond this curve, everything is impossible, right? So these areas will be too good to have because the optimization direction is towards the top left. But the lower right of that curve, everything is possible. So the feasible region is huge, really. So every point is a sequence, 2.4 times 10 to the power of 632 candidates. And all these candidates left to this line are uncharted region. Like even Moderna people didn't explore because they didn't have a way to optimize, to do the combinatorial optimization of energy. So just to summarize, mRNA stability is a major challenge for vaccine development, and if we can solve it, we can reduce the cost, and we can reduce the dosage, and to make it more usable for the general public. And we proposed efficient algorithm for optimized mRNA vaccine design. It turns out this problem was solved 60 years ago. We just rediscovered it, and we can design very long mRNAs for the spike protein in just an hour. By the way, this is also the target for Moderna and many other vaccine companies, and the codon autoality can be jointly optimized in this two-dimensional way. And this is useful not just in vaccine design, but also in like antibody design, cancer treatment, gene therapy, synthetic biology, and so on and so forth. And since the release of our preprint, several major vaccine companies from the US, EU, and China have been in contact with us, and some of them are doing the wet lab experiments to verify whether our designs, in silico designs, are actually useful in practice, like in vitro and in vivo. So those are wait to be seen. So we are only doing a hypothesis. So with that, I would like to thank you, and please try our web server and our preprint here, so you can upload any protein sequence, and within this very short time, we can return you the best vaccine mRNA vaccine sequence. And these are our co-authors and collaborators, and our General Manager of Battery Research, Julia Lee, and we thank Professor Vijay Das from Stanford for giving us this problem to work on. I would like to thank you again for your attention.